Morning, everyone, and welcome to Kudo Biotechnology and LineaRx Applied DNA's Tech Talk and Lunch and Learn. I'm Molly McLaughlin. I am the CEO and founder of Kudo Biotechnology, and I welcome all of you here in person and those of you who have joined us from online. So today we're going to be speaking about the optimizing of mRNA production using a novel synthetic DNA platform for rapid manufacturing of mRNA medicines. We've been working on this project for almost a year now, and from early experiments to where we are today, it's a great set of data, and it's very compelling to our personalized medicine model that we have built for rapid manufacturing of personalized medicines from sequence all the way through drug back to patient in a very short period of time, which we'll get into in detail. So with that, I would like to introduce you to Kudo Biotechnology. So Kudo is a all-encompassing full suite of service CDMO. We have been around for about 14 months now. We were carved out from a mRNA infectious disease company who had a failure in the clinic. And so I was hired to carve out the GMP assets of that company and create a CDMO. With that, we have created the CDMO that's multi-product, multi-purpose. We have 2 sites. We are a global player, so our main site for GMP manufacturing is in Shanghai. And then here in the Boston area in Needham, we have a 5,000 square foot process science center of excellence, where we do everything from plasmid, DNA process development through IVT reaction development, analytical development on all those, and then also the LNP encapsulation development and formulation for fill finish. So that's all done in the Needham here in the Boston area offices. We have global customers. We manufactured under GMP and release clinical product in multiple countries globally. And so we have a pretty strong track record. The key difference is that we do it all under one roof. And we are currently doing that with plasmids. And then now with the LineaRx technology, we're looking to add this capability here in Boston to be able to do all of this under one roof with the synthetic biology template versus plasmid, and then IVT, LNP, and fill finish here in Boston. So my management team is seasoned. They either come from mRNA or they come from CDMO. And so we do have a depth of about 350 man-years of experience in this space, everything from plasmids to IVT to LNP, and then also the formulation development and analytical development. This team comes from the usual suspects in mRNA, but also the usual suspects in CDMO as well. So big players such as Catalent, such as Lonza, such as BioVectra, Avicia, etc. We're also adapting advanced technologies such as applied DNA's technology in LineaRx, but we're also have a portfolio of patents in other innovation within the LNP space for lyophilization formulations. We also have some patents in the areas of UTR and codon optimization to increase protein expression of your mRNA. So we do have a lot of innovation in this space, and we're adopting those for manufacturing. They are mostly focused on process and then also delivery. In adoption of that innovation, we are really ensuring that we are best in class as far as any of the target product profile for your products. So when you look at performance and you're looking at IVT efficiency, we have greater than 200 grams mRNA to grams DNA. Our capping efficiency is traditionally and typically 98%, and that's using like a capping analog, and we also have an enzymatic capping capability as well. mRNA recovery in the 89%. We're always constantly trying to get that higher, and then encapsulation rate's 95%. From when you start looking at the LNP particle size, It is consistently on the target. We also have our polydispersity index is incredibly low compared to the industry standards. When it comes to safety, endotoxin bioburden, sterility, we're best in class. We're well below industry standards and requirements from the FDA. And then if you look at the mRNA integrity, it's also 95% typically is on average in our hands, not only in small scale production, but also in a larger scale GMP production. The big key is for us right now with plasmids, because we're all under one roof, we're at about 6.5 months from the time you send us an email with your sequence in it to when we're able to produce GMP fully released product. That dramatically changes, however, with this new technology. With that, I'd like to introduce Aaron for LineaRx, his portion of this session.
Thank you, Molly. I'm here with my boss, Clay, here. So if you guys have any technical capability questions, I'm going to direct your question to Clay because he's much more experienced on that regard. A little bit about my background. I was trained as an engineer, but I spent the last 6 years working on this RNA deliveries. So I have a little bit of experience on this IVD process. And I also want to thank in advance the Kudo team for generating all this fantastic data. They really spent a lot of work and time on this. So thank you. I'm just going to let a couple more people in. So just a quick history regarding LineaRx. LineaRx is a subsidiary of Applied DNA, which is a NASDAQ-listed company. We have significant IP estate on our PCR-based process and also the proprietary enzyme that drove our production. We also have a GMP manufacturing facilities coming online very soon, so keep an eye on that. So, like Molly said, we specialize in large-scale PCR-based DNA manufacturing. And our process is actually quite straightforward. So the customers could either provide a digital DNA sequence to us or a physical template. We'll design the primers and run a small-scale study, confirm everything is working, and then we'll amplify the sequence in large scale and do all the analytical testing on the amplicon and ship the product back to you. The process is highly scalable and well-suited for GMP. And our PCR process is actually particularly suitable for IVD template because it only utilizes one single high-performance DNA polymerase in the reaction, which reduces the cost significantly. So in one single enzymatic process, we can incorporate uniform poly A tails, which is a big problem in plasmid-based production, because plasmid production tends to truncate the poly A tails over the cell cycles. So in this particular examples, we're using 120 nucleotide poly A tails, but we can offer up to 180 nucleotide per product. And we observe very consistent RNA integrity. As Suku would dive into this a little bit more detail later. So obviously, we supply regular DNA, but we can also attach chemically modified tags on a DNA template. PCR make it really easy to do this, which we'll later learn when paired with our proprietary fusion RNA polymerase. It would drastically improve the RNA quality. So PCR allow us to manufacture DNA very fast, and we can typically make milligrams of DNA in 2 weeks and grams level of DNA within 45 days. So this is very useful for clinical and commercial workflow that requires a quick turnaround time.
And can I ask, is that GMP in 45 days?
Right, so 45 days is the DNA, the synthetic DNA, and working with Kudos, we're also working on our mRNA workflow.
But yeah, just to answer that, so yeah, we can right now we are building GLP products. We can go from right now a existing template such as a plasmid to about 10 to 15 mg scale in 14 days in our current facility. We can go from synthetic, an email with a synthetic build into amplification, that same mass in about 21 days. As Aaron said, we're building a GMP facility. That facility is coming online in about 2 or 3 weeks. There, our goal is existing template to 10 meg scale in about 21 days.
Very good. Thanks, Mike. So back to linear IVT. So we also have a unique mRNA production platform we call linear IVT. The two key components for RNA synthesis are the DNA templates and the RNA polymerase. In the ideal world, we want to have the RNA polymerase synthesize the RNA and leave the RNA product alone. But many scientists and great researchers later found out that RNA synthesis is far from ideal. So double-stranded RNA was produced as a byproduct during the IVT in vitro transcription process. This is a huge issue because cells often recognize double-stranded RNA as a viral infection. And even a small amount of double-stranded RNA can shut off the protein translation machinery, which would undermine the efficacy of the vaccines. Double-stranded RNA is generally formed through a process called self-templating, which happens when the RNA polymerase kind of started to bind to the RNA products and extend itself on the 3' end. This resulted in an undesirable RNA-dependent RNA synthesis process, which created the double-stranded RNA. More importantly, double-stranded RNA is also notoriously difficult to purify because the double-stranded segment is usually around 50 bases long, so it's really difficult to separate, for example, a 5,000 bases RNA target from a 5,050 bases double-stranded RNA molecule. So what we are doing here at LineaRx and Kudo is to design a system so that only DNA-dependent RNA synthesis will proceed. And more importantly, we want this platform to be directly integrable to the existing batch IVT process, so that scientists can use our platform without any additional instrument or microfluidic device. Our IVT platform is unique in the sense that we have a rational approach to address these double-stranded RNA impurities. This is different than any approach done by others using directed enzyme evolution. So the first thing we do here is that we leverage our large-scale PCR platform to incorporate modified chemical tags on all our DNA. So this can be added through a one-step assembly process, which also incorporates the poly A tails that we talked about just like a few minutes ago. Here, we show that the chemical tags of our modified DNA would bind to the DNA binding domain of our proprietary fusion RNA polymerase for RNA synthesis. So a lot of people might have concern and think that maybe if the DNA was bound to the fusion RNA polymerase, it would be difficult for the polymerase to move along the DNA template. But we have verified that this is not the case. And in fact, our linear IVT system can make a wide range of RNA lengths without much issues. And the yield that we observe across different template lengths are consistent, if not better. And what distinguishes our platform from everyone else is that our buffer is crucial in making this platform work. Our buffer system contains some proprietary additives to disrupt the molecular interaction between the nucleic acid and our RNA polymerase. So normally, in a wild-type T7 system, this would not be a good idea because when you're adding things that prevent the polymerase from binding the DNA template, you will reduce the RNA yield. But our system works under this condition because the fusion RNA polymerase are physically tailored to the modified DNA template. So what's more, these additives are tunable. You can dial up and down the concentration of the additives to achieve optimal results based on your requirement. So for example, you can dial up the additives when the customers require less double-stranded RNA in their final product. And you can dial down the additives when the yield is more important. The additives would make it difficult for the RNA polymerase to bind to the RNA products and proceed with the self-templating, which we just described earlier, that could result in more double-stranded RNA. So just going back to this slide for a summary, we are preventing double-stranded RNA formation upstream in the IVT reaction by using buffer additives to discourage double-stranded RNA formation through our tailored modified DNA and fusion RNA system. And through this system, we also encourage RNA. So from a process development point of view, less double-stranded RNA means a more simplified downstream purification process. So we can turn around higher quality mRNA much faster. So now I will pass the mic to Asuku. She and her team have done a detailed DOE study on both of our DNA templates and the linear IVT platform. Asuku.
For explaining the DNA template and then the proprietary T7 RNA polymerase. So I'm Atsuko from, can you hear me? Speak up a little bit, please. So I'm Atsuko from Kudo Biotechnology. I'm director of the drug substance process development. And so if you have any question during the presentation, just feel free to stop me because that's the way more interactive and then I feel comfortable that way. So Kudo is like an RNA focused CDMO, like Mori talked about during the introduction. So as a CDMO, like we notice, or like everybody noticed, after COVID pandemic, the next most active area for RNA is like a personalized application, like cancer therapy, personalized cancer vaccine, or cell and gene therapy. So for these applications, the batch size is usually smaller. You don't need like millions of doses like vaccine application. And also the expected turnaround time is much shorter compared to traditional vaccines. So still like RNA technology is like good for pandemic responses, infectious diseases, but more people are interested in cell and gene therapy or cancer vaccine type of application. And then we get a lot of questions about that. And we see the common challenges from those potential, like for those companies who's making that kind of therapeutic RNA, like a template DNA is usually a bottleneck because it has a long lead time. So they need to, or you need to select the clone, right clone with like 48 tails intact, and then you have to do the fermentation, scale up, time consuming equalized selection and cell bank generation. And you have multiple banks, you need to release all the equalized banks with like a lot of testings. And then of course you have to store it in GMP. Many things are at cost. 48 instability, like Aaron mentioned, and batch to batch inconsistency is like issue. In addition to the DNA template, controlling dsRNA impurity is like they ask the question to us, and then like they want to have therapeutic RNA, how low it should be. No one knows the answer. It's very product dependent. What's your dose? How many injections you need? And then no one really knows the answer to the correct specification. So Kudo Bio and LineaRx have developed a workflow to provide a solution for that kind of questions and challenges. So Kudo provides, LineaRx provides 100% enzymatic cell-free DNA very quickly, and then we can do the RNA and LNP formulation under one roof. So it's being a one-stop shop. It reduces the timeline. And so we target like in total 6 to 12 weeks turnaround time. So if this like modified DNA template and also unmodified DNA template, are they like working? So we did the feasibility study for this synthetic DNA. The first study we did is the feasibility study using the unmodified synthetic DNA. So this is like a very like a simple direct replacement of the E. coli template. So we use the COVID vaccine as a model antigen, because Kudo made the GMP batch of this COVID vaccine successfully, and then it's released for clinical trial. So we have like high quality process and benchmark RNA. So LineaRx produced a synthetic version, PCR template using the same sequence. And so we did a simple head-to-head comparison of these two. So only difference is pDNA needs to be linearized and purified. And then we use the industry standard T7 wild-type polymerase. So RNA is purified by oligo-DT chromatography and then tested for various quality attributes. So here is a summary of the comparison. So on the left, you see the RNA, like a chromatogram for fragment analyzer, E. coli DNA, and comparing on the right side is PCR-based template. So you can see very clean peaks for both RNAs. Integrity is over 93% with E. coli, comparable to synthetic DNA. Yield was exactly the same in our hands, and capping efficiency was much higher with PCR DNA template. The dsRNA level is not shown here, but they are both comparable to Kudo's GMP release batches. So it was very clean, especially, at least it's gonna meet the spec for vaccine application. So this is another data from the feasibility study. This is HPLC purity assay chromatogram. So you can see E. coli DNA has somehow two peaks, but we didn't observe this one with synthetic template. This was very curious because we have observed this two peak issue during the GMP campaign, and then we couldn't really tell why it's showing two peaks, and then fragment analyzer gives you very clean peak, but you can tell RNA is not as uniform as you can think from fragment number. If you run HPLC, you can see more different type of species, but I can tell synthetic DNA makes more uniform structure with same sequence. So the next study is I'm going to move to the modified template with LineaRx's proprietary RNA polymerase. But if I go to the one, does anyone have any questions? Okay, so this is the same COVID RNA construct, but just made with different ones. So this is the modified template. It's supposed to work with this Aaron just mentioned, the special T7 RNA polymerase. So we did the IBT with our platform, our LineaRx's platform process, and then RNA purification, and then we confirmed the RNA quality was comparable to the previous one. So then we started thinking, can we improve the integrity or yield, or what kind of parameters impacts the RNA quality in this platform? So we did the 2 rounds of DOE study to optimize. So IBT optimization with DOE is very standard approach because RNA, we call it platform, but it's very different from construct to construct. The lengths or UTRs or secondary structure, you don't really know until you do the IBT, what kind of yield and purity it gives you. So we offer this kind of DOE IBT optimization study to the clients, and then we applied it to this linear IBT platform. So for the first DOE study, we tried 4 different factors: RNA polymerase concentration, template DNA concentration, NTP concentration, and cap panel concentration. In the second round of DOE, we did further analyze the effect of template DNA input, cap analog, and also we added the KCL as the factor because this RNA polymerase is very unique. It can take higher amount of salt, and then it contributes to some quality attributes. responses we tested is the same, like RNA yield, integrity, and dsRNA content by dot plot to test a lot of many samples because DOE requires like a lot of samples. But after that, we pick some conditions, do the confirmation run with full-scale oligo-DT purification, and then like the dsRNA was measured by more quantitative way. ELISA, capping efficiency, and most importantly, cell line responses tested by LineaRx team. So this is the first round of IVT. I mean, I can just show you some example data here. So green bars show yield in each individual condition, and then these pink dots are integrated. So you can see some conditions gives you like high yield with high integrity. Some of them don't really perform like that. Half the yield, but still integrity is high. So we also here is the dsRNA content, like high, medium, and low, because dot plot is semi-quantitative, so we don't have the exact number. So we put these responses to a statistical software, JUMP, and then the JUMP model, like what is the critical factor for these conditions. So from DOE plus one, we identified a few conditions that improved RNA yield compared to the first initial conditions developed by LineaRx team. So it was still work under progress. We improved some attributes. Integrity looks like around 75-80%, but this is please remember this is not really purified with oligo-DT. This is a quick control experiment. So this number can be, I will show you the data later. Also, dsRNA level was comparable but slightly lower to the RNA produced by conventional E. coli-based IVT. So it was already promising. dsRNA level is low to start with. Then we throw in some other factors, especially the KCL. So you can already see in the second round of DOE, yield is in general lower, because maybe Aaron just mentioned, you can dial up and down many factors in this platform to bring the integrity up, or it can sacrifice some yield, but dsRNA can be that kind of stuff. So if we modeled it with using this data, so I can show you the prediction profiler of the IVT performance. So from 2 rounds of simple DOE, we could make this prediction performance profiler. So you can see the KCL condition. I think many people are familiar with this, but if you see the slope is flat, that means it's not really significant impact. If this slope is short, that means it's impacting this factor a lot. So KCL has the impact of integrity in RNA. DNA template input, if you increase it, yield goes lower. You can play with this model to predict the conditions for your construct. So we picked some conditions and then did the confirmation run with oligo-DT purification. So here are some conditions we tried. So optimized by DOE, like a JUMP prediction model. If you go back to this one, this is the best condition the software thinks. But green gap concentration was 2 millimolar. It's very low compared to the industry standard protocol. So so that's why we included next condition 4 millimolar green gap, and then according to this prediction profiler, it shouldn't impact any other attributes too much. So we are comfortable to use this one. And also this is the original protocol. This requires a higher amount of green cap. That's not something we want, I believe, because it's very expensive. So we have 3 levels of green cap. Then we did the E. coli template with wild type D7 as a benchmark. So as you can see, linear IVT platform has higher in the conditions compared to the E. coli. And then RNA integrity number was around 90%. Sorry, this one is missing, but this is very fresh data, so we are still analyzing some samples. But overall integrity was 90 or higher than that. And then capping efficiency was like as we expected. 2 millimolar green cap didn't give us good capping efficiency, but with the standard concentration of 4 millimolar, it's over 96% with higher capping analog concentration. It's near 100% with this platform. So again, I'm just stressing Aaron's point. This platform is very easy to customize for the construct, and then what's the most important factor for you, we can dial it. And then rsRNA impurity by ELISA. So E. coli DNA process as 1.0, it's like all the conditions were lower than that, especially this one is significantly lower. But please note this E. coli DNA process gives you low dsRNA because we optimize this one for clinical batches. So like comparison is like maybe too harsh here. So this is a cell-based reporter gene assay to predict the innate immune response stimulation. So we use this data was generated by LineaRx team. So this is the dual reporter gene cell line. So it's gonna stimulate it by dsRNA, activate TLR3 pathway, and then it's goes to NF-kappa B. So this is SEAB in pink is like NF-kappa B dependent transcription, and then the yellow one is another showing ILD promoter dependent responses. So as you can see here, like, compared to the E. coli process, linear IVT RNA products show less immunostimulation in cell-based assays too. So this data is correlating very well with ELISA. So here is a summary of our study. So we just summarizing everything. LineaRx offers two different PCR-based templates. One is unmodified synthetic DNA. This can be direct replacement for E. coli DNA. So we proved the comparability to the current process with E. coli with wild type T7. And then this can be done without a lot of optimization. So this is cost-effective, time-saving replacement for the industry standard process with E. coli. And then interesting, like this linear IVT platform is a novel platform. It's all cell-free, designed for LineaRx's proprietary T7 RNA polymerase. It produces RNA with lower dsRNA impurity without sacrificing yield and integrity. So this, we believe this is the ideal platform for therapeutic applications, requires high dose or like a repeated dosing because of this mitigated dsRNA content. So this is the last slide I have. So Kudo provide like a complete suite solution from patient sequence to the point of use for the patient. So this is very good platform for personalized application. We can do tailor-made RNA-based personalized medicines, rapid supply of clinical trial material as 12 weeks from sequence to full release, and then patient-specific manufacturing in small batches because this is like, again, this scale is very flexible. We can do small batch to like or scale linearly. The manufacturing will be available in US and also it's already available in Asia. So that's all I have. So if you have any questions, I'm happy to answer.
Because I have a question. Sorry, a comment. you made the comment, I think it's on slide 35, that this is your GMP assay, right? So this is a very well characterized IVT reaction, right? So
Yes.
a lot of dsRNA data that's out there is using very dirty reactions to then reduce the dsRNA, which you can do with wild type. But why I think this is so interesting is that this reaction has been optimized by you guys over a very long time. This is a GMP low dsRNA reaction to start with, and that's our comparator. So what I think is so interesting about this data is that these are the DOE studies. There's still further optimization that can happen, but we still have lower dsRNA than the E. coli optimized GMP. So I just wanted to point that out because I have seen a lot of data out there in the industry where their comparator is very high dsRNA, and you can certainly do that.
Yeah.
But this is a very good reaction to be comparing against.
Yeah. Thank you. I wanted to stress that point. Yeah. So this is like optimized. It's very fair comparison, but it doesn't look amazing maybe. And also I didn't want to use the number because many different kits or different antibodies for ELISA, it gives you very different numbers. So you see numbers on some companies' slides, like a slide deck, but you don't really know what you're measuring or comparing apple to apple. It's not the case most of the time. So this is what I can honestly compare with my product with consistent performance of the ELISA.
Thank you. A really great job.
Aaron, we actually had some questions come in online regarding the PCR-based DNA production. Should there be concern about the mutation rates?
Yeah. So I think like usually like for Clay, like jump in if you can. But I mean like usually for like plasmid, I think the mutation rate is around like 10 to negative 7, and polymerase that we're using is say like -6, -7. But I think one thing to note is T7 itself usually have a mutation rate around 10 to -4, -5. So even though sometimes the polymerase might be slightly produce slightly higher mutation rates than like a plasmid process, but the wild type T7 is actually much worse than compared to the DNA polymerase.
It is, yeah. And just to add to that, I mean, plasmids are not perfect, right? There was a recent Nature article on issues with plasmid and the lack of sequence fidelity with those. We've looked very hard at the fidelity because that's an obvious, criticism that you could have a PCR. We've done a couple of things. One, we have a robust NGS program around that, so all of our products are NGS verified. But two, we've developed an enzyme with an enzyme manufacturer along with a proprietary the reaction buffer. So, we have gone through not only NGS studies, but also cell-based the DNA sequence fidelity studies, and we're very confident about the sequence fidelity. We've also, to Aaron's point, looked at the RNA through RNA-seq, and again, we have been unable to identify any errors as compared to the pDNA, the template. So we're very confident about the error rate.
Another one was, how is the poly-A added to the pDNA template? Is there a limit for poly-A length?
Yeah, so right now, I think we're offering up to like 180 poly tails. Through our PCR process, we incorporated directly during the amplification process. So, like unlike like in a plasmid setup, like usually through cell cycles, like if the cell doesn't like the poly tails, it will start truncated through like evolution. But in our process, we do it in a cell-free manner, so it does not go through any cell-based process, and we integrate directly to our DNA template. So that's why we have a pretty uniform poly tails when compared to plasmid.
Another one here with, which cap analog does the proprietary LineaRx polymerase work best with? Have you tested with different cap analogs?
Yes, I believe so. Like we have done a clean cap, we've done like the Archer cap. Have we done any more?
That's it. We're looking at other cap analogs, but as an overall trend, and of course there's outliers, but overall trend, it does seem to lend to very high capping efficiencies. We're actually doing a study to look at that now, but overall the trend is that it has very high capping efficiencies.
Jessica, do you have anything to add on the capping analogs?
Yeah, actually, we at Kudo, not with this LineaRx template, but we did a lot of a cap analog assessment because they are in the market, they're more clean cap similars or a cap analog with unique modification and then IP free. That's what they say. So we are interested in finding alternative capping reagent or compare performance, and then actually we see a big differences. It was surprising. So some cap analogs works very well, but some of them in the market in our hands 80% max. So there are very a big difference in terms.
For Kudo, what's your smallest and largest GMP batch size for mRNA?
RNA, we can do 100 mgs because we can do many small, we can do even smaller scale, couple of mgs for research with representative scale, but for GMP, you need a release and stability and stuff. So I think it could be 100 mg RNA scale, LNP accordingly. That's what I was actually at the conference yesterday, some symposium, and then there was big pharma doing a personalized cancer vaccine. They say that their batch size is around 100 mg. Turnaround time is 3-4 months. So I think our platform is much faster.
And we also think a PCR front end to that makes a lot of sense, right? Because while PCR can scale to very large, it excels at very rapidly making, between 1 and 10 mgs of template, and that's what you need for that batch size. So we think it's a great combination.
Yeah, so they say there are bottleneck is of course the DNA takes a couple of times, couple of weeks. E. coli and the release is at the end. Product release is taking a long time, but in our case, Kudo has everything under one roof, so release is quicker.
Any further questions?
If there's no online questions, we can take questions during the lunch.
Absolutely. And I do want to thank Josh Syme. He was the project manager on this event. I think we have 30 plus people online, and quite a good crowd here in person. And so thank you, Josh, for all your hard work. Kathleen Brackett also assisted. Aaron Atsuko, please, let's give them a round of applause.
Yeah, and also shout out to Morgan in the back of the room. She's scientist in my
Hey,
team.
Malcolm.
She did all the hands-on work.
She did all the hard work.
Yeah, I'm just presenting. Thank you.